There we go. So the title of my talk is A Quest for Treatments for Pancreatic Diseases. And I probably should retitle it a little bit from what you're going to hear me talk about is the quest for improvements in outcomes in pancreatic disease, because some of the things I'll talk about is how we can make diagnosis early, for example, with pancreatic cancer. And I saw that Julia Meyerly talked about that a little bit earlier and talked to you about how we're trying to do this in the United States. So the talk I'm going to give is surrounding a consortium we have in the United States called the Consortium for the Study of Chronic Pancreatitis, Diabetes, and Pancreatic Cancer. And as I get into it a little bit, I'll explain a little bit how those diseases are connected and how this consortium came together historically. I lead this consortium with Chris Forsmark at the University of Florida, and the consortium is managed by our National Institutes of Health with two of our sub-institutes, the National Cancer Institute and the National Institute of Diabetes and Digestive and Kidney Diseases. From those institutes, Joe and Renato from NCI and Jose Serrano from NIDDK are overall managers from the National Institutes of Health. So hopefully I can make the slides go forward. Well, that's something I'm having trouble with today. I have nothing to disclose. First, I'll describe how our consortium is operating. We have multiple working groups, and you can see by the names of them the kinds of topics they work on. For example, we have a diabetes and PDAC, pancreatic ductal adenocarcinoma working group, and they work on a project very similar to what Julia described that's been ongoing for 4 or 5 years now, called new onset diabetes. And those are patients greater than 50 years of age. In the United States, we're recruiting 10,000 patients to that cohort to identify biomarkers and liquid to make an early diagnosis. And I'll argue a little bit later about how that's going to potentially improve outcome. We have a working on chronic pancreatitis, and that is ongoing, and we recruited 1,570 patients, and most of those are already recruited. We have a pediatric working group, and my son is actually part of that, and so is Sohail, and they have a working group where they're recruiting 600 plus patients with acute relapsing and chronic pancreatitis, and a study we called DETECT from our diabetes type 3C working group. And those are patients who have either pancreatic cancer or pancreatitis, chronic pancreatitis with diabetes, and it has studies designed to determine the mechanism of the diabetes. That's just an overview of working groups. Those working groups then have cohorts that they're following, as I've described in this slide, and those cohorts are important for describing determining the natural history of these disease processes. But each of them also develop multiple ancillary studies. And again, I'm hard. And oh, there we go. Ancillary studies. We have about 6 of those now. And those studies are to those cohorts and other patient clinical cohorts to develop liquid and imaging biomarkers for early diagnosis and identifying and distinguishing the different pancreatic disease types that I listed in the title of the consortium, development of drugs for prevention and treatment for each of the pancreatic disorders, considering their complex interrelationships, to conduct clinical trials for validation of biomarkers and prevention treatments, and to collaborate across multiple disciplines, scientists, clinicians, healthcare organizations, nonprofits, our FDA, Federal Drug Administration, and commercial entities to basically improve outcome in these diseases and their interactions. This is a slide just demonstrating the interactions that you're all well aware of. And on the outside are risks for pancreatic diseases, digestive enzyme mutations, but also ductal cell mutations, alcohol and smoking, diabetes and obesity. And this slide just shows that in some patients, there is a continuum from acute pancreatitis to recurrent acute, chronic. Some of those develop diabetes. And by the way, some after acute pancreatitis develop diabetes as well, which I didn't indicate here. And then a portion of these patients go on to develop pancreatic adenocarcinoma. These are risk factors for pancreatic carcinoma, adenocarcinoma, but as you all know, they're not the only risks for pancreatic adenocarcinoma. This is just a high-risk group, but doesn't represent all of the cases in the disease. One thing I wanted to bring out in the interrelationships shown in this slide. And what you see is there are double arrows going between each of the disorders. That is pancreatic cancer, pancreatitis, and type 2 or type 3C diabetes. That is diabetes associated with exocrine pancreatic disorders. And so that shows you the complexity. But one thing I was going to point out is that, and it's a clinical finding that we're using to try to identify pancreatic cancer patients early, is that those patients who have pancreatic cancer actually can present with diabetes, and we would call it type 3C diabetes. It could be one of the earliest clinical markers. Now, obviously, that's complicated because the majority of patients who develop diabetes are going to have type 2 diabetes, but it is one clinical indicator, and that is the basis for developing the cohort study that I described, where we're recruiting 10,000 patients who have diabetes after age 50, because those patients among them have a small number, about 1% to 2%, that go on within a 3-year period to develop pancreatic cancer or have the diagnosis of pancreatic cancer. They probably have it at the time of diabetes detection. So I'm going to talk about two kinds of projects that I'm more involved with. One is early diagnosis of pancreatic cancer, and the other is potential treatments for chronic pancreatitis. So this slide was published by the Mayo Group, Suresh Chari et al. In 2015. And what it tries to show is that if we make the diagnosis of pancreatic cancer earlier, we can improve survival. So what you see on the top line are the current proportions and what proportions of the stages of the disease that you would need to quadruple survival. So it points out, as you see at the bottom of the slide, the average 5-year survival currently is 4.1%, and 4 times that, or quadrupling it, would be 16.4%. But the important point of the slide is that in order to achieve the 16.4% based on outcomes from patients diagnosed at certain stages of the disease, you would have to increase those patients with early diagnosis where there is a surgical potential long-term survival. You would have to go from those that are surgical cases that include the top 3 stages. You would have to go from a total of about 20% there to more like 50%. So that shows that we really have to, one way that we can improve outcome is to improve early diagnosis. So taking that into consideration, I come back to the point about using hyperglycemia or diagnosis of pancreatic cancer based on hyperglycemia. And this was published again by the Mayo Clinic group in the United States, with Sharma and Chari being the leaders in 2018 in the journal Gastroenterology. And what they point out, if you compare the glucose trajectory in cases, which are the blue boxes, compared to controls, and then you go backwards from the time of pancreatic cancer diagnosis, that they start to diverge 36 months earlier in their glucose levels. Now, again, these patients are most likely going to be diagnosed as a type 2 diabetes just because the number of patients that have pancreatic cancer in that group are small. However, this shows in those who do go on to pancreatic cancer, there is a trend line of increasing glucoses. Another important feature that was demonstrated by the group at Mayo, but also by Christy Joan, who works here at Cedars, who's an epidemiologist, is that there are other factors that you can rely on to identify high-risk populations. And I'm going to focus on the middle set of bars. And what this is, what this represents is the 3-year period before diagnosis. And it shows that compared to patients who have no weight loss, there is a significant increase in risk in those who do have weight loss. So going to the second set of bars, there's a blue, orange, gray, and yellow. And you'll see that the different colors represent different amounts of weight loss. The blue, the greatest amount, greater than 15%, and the yellow, the least, less than 2.5%. And what you see is that the likelihood of developing pancreatic cancer in those who have diabetes and are taking insulin, that the risk is 8% in those who have weight loss of 15% or greater. So it shows that there are formulas, and actually Dr. Chari and his group have developed formulas with other risk factors, this being one of the predominant ones in identifying patients with early pancreatic cancer. So if we have a patient who has some weight loss and develops diabetes, we should really think about the possibility of pancreatic cancer. So this is actually almost a duplicate of a slide that Julia showed. And it goes back to the concept that we can identify a group of patients at risk with new onset diabetes, but only about 1% of that group is going to go on to have pancreatic cancer diagnosed within 3 years. So biomarker tests, liquid biomarker tests, can be a first screen. They have to have significant sensitivity and specificity, as Julia pointed out, but that would decrease the or increase the enrichment of the population to more like 1 in 15. And then there can be further screening with CT, for example, or other imaging techniques, et cetera, to improve the prediction even more. So in this case, it was 1 out of 3, just the concept. So taking that into consideration, we are also working on a liquid biomarker test and a imaging test that I'll describe briefly here. So we're working with Tanya Yerkovich, who is at St. Bart's in London, who's developed a 3-protein urine biomarker test. And this just shows where we are in the process of developing the test. We've gone through clinical validation in a smaller group of patients, about 300. We're near completion with a larger validation test, and we plan to launch that test in the third quarter of 2022 as an LDT, that's laboratory developed test, but also we're developing a kit test. And so what is this based on? This is based on Tanya's work at St. Bart's, but using samples from Cedars and other places. And she's developed these 3 proteins, LYVE1, REG1B, and TFF1, that she identified first with discovery in urine proteomics, and then validated it in groups of patients. And the groups of patients, as you'll see, are listed on the bottom of each of these biomarkers, are healthy controls, H, benign exocrine disease, BE, PDAC at the earliest stages that I said earlier are surgically amenable, to the later stages, as you can see at the last bar graph here. And what you see is that looking at the early stages of pancreatic cancer, which is the 3rd bar from the left, and each of these is that we have a signal that distinguishes those patients with early pancreatic cancer from benign disease and healthy controls. And furthermore, I won't show this here. Furthermore, what we've demonstrated that there is, we can improve the sensitivity to plus 90%. And with that sensitivity, we have a specificity of about 85%. We can improve that even furthermore by adding CA19-9 to the assay. So that's one kind of test that we're developing that goes along with what I showed our consortium is trying to do. And there are other tests that companies and individuals are trying, and also Julia mentioned what they're working on. The next thing I'll describe is prediction of early diagnosis of pancreatic cancer in CTs based on artificial intelligence or machine learning. Doing that here at Cedars with a bioengineering group, UCLA and Cedars-Sinai bioengineering group. The key scientist on it is Tusif Qureshi, and the leader is the bio lead. What we did is took advantage of a known published fact, is that we miss pancreatic cancer in radiological examinations, probably. And the reason we know that is that there are patients that show up with the diagnosis of pancreatic cancer that have had previous CTs for different reasons where the diagnosis was not made. So those CTs happen within mostly within 6 months to 3 years beforehand, but some even longer. And so the question is, are there signals in those CTs that that maybe we're missing, either because of how would you call it, the acuity of the radiologist, or because the signals can't be seen no matter how they're represented to the radiologist. So what I'm going to show you is just a brief example of how we use AI. So this just shows a pancreas from a healthy control patient. There's a lot of processes that goes into this, but it's basically segmenting out the pancreas with automatic algorithms. And we also have algorithms that segment out different portions of the pancreas, including the pancreatic duct and the different head, body, and tail. And if this works, you can actually see that we can rotate this in 3 dimensions. But again, this is an artificial intelligence representation of a segmented pancreas, and it's our normal representation. The next thing, as I'll show you, is a pancreas where biomarkers are identified by the red spots that are different from normal, and were identified in a group of patients that had CTs done without the diagnosis of pancreatic cancer. So we can apply that a machine learned algorithm to CTs in prior or pre-diagnostic scans and identified areas of inhomogeneity. And we have names for those. We call this a feature map. And this is an example of one showing tissue heterogeneity. And that area is the biomarkers that are different from normal that come from our study of multiple cases. And this is also showing that there's more uniformity in this part. We do recognize that there might be changes in the non-cancer part of the pancreas that are due to the cancer, and that'll be one of the findings we'll make. So we're in the process right now. The National Cancer Institute has allowed us to go forward with resources to recruit several thousands of scans that are pre-diagnostic, diagnostic, and controls across multiple medical centers in the United States to perfect might not be the right word, to continue to improve the algorithms so that they can be applied clinically. And then the last thing I'm going to talk about is our interest in chronic pancreatitis. Obviously a very difficult disease, has been talked about earlier today. And we have both biomarker studies, but also developing or at least initiating trials for treatment of chronic pancreatitis. And we have, I would say, about 5 that have been initiated in the United States with different concepts driving the agents and the treatments that have been chosen. I was going to show you one that started in Los Angeles with Beecham Wu. Beecham is my colleague at Kaiser Permanente, Southern California, which actually is just like a 15-minute drive from where we are in central Los Angeles. He conducted a retrospective longitudinal study from their data from 2006 to 2012. And the main finding was that those patients who took simvastatin had a markedly decreased incidence of pancreatitis. And you can see there's some dose dependency on it. That was published and has gotten widespread interest and actually validation by others. But we thought, well, is there a mechanism that we can use to understand this a little bit better? And so we started working on it. And I'll just show you a few concept slides about our work, and we published this as well. And it comes from the cardiac literature. So one of my colleagues here at Cedars-Sinai in Los Angeles is Roberta Gottlieb, who's a expert, world expert on mitochondrial function in cardiac diseases, and especially interested in mitophagy. In other words, how does a cell get rid of reactive oxygen species producing a leaky mitochondria in order to prevent them from initiating inflammatory and other cell injury responses. And she demonstrated that statins actually can have an effect on these processes by improving mitophagy function in cardiac ischemia-induced cardiac risk tissue for necrosis. So we thought, well, can we apply that to pancreatitis? So this is just a general figure about how a cell deals with sick mitochondria. So it shows that if you go from the left to the right, that there is a signaling system involving PINK1, parkin, ubiquitination, P62 translocation, and then ultimately delivery to autophagosomes and actually digesting up the disordered mitochondria. So she's developed techniques to follow mitochondrial turnover, and this is just a picture of that on the right, just for color. But what we're able to show using this cartoon is that this process of mitophagy, where a mitochondria is taken up by a double membrane structure leading to an autophagic vacuole, and then proceeding to degradation, that there is a process where the autophagosome, what do you call it? It fuses with the lysosome to deliver digestive enzymes for degradation. And in pancreatitis, this step by several people in our field, including Anna Gokolskaya and Ashok Saluja, who's sitting there in the audience, is blocked in pancreatitis. And that allows for the accumulation of undigested material. So what we found is that that block was reversed by simvastatin. And we did several measurements, but one was just following these different structures by electron microscopy and trying to grade or score where the process was started and what the effect of simvastatin was. And what we are able to show is that that simvastatin reverses this block that I show here, and that the mechanism actually is something we're working on now that we think we can address, and that would take away the block and improve mitophagy and potentially improve outcome. We think that's how statins can work. And so finally, I'll show you that we started clinical trials with simvastatin. We started in Los Angeles, believe it or not, and we weren't able to recruit enough patients. The trial we designed was too limiting. We were requiring endoscopy and pancreatic fluid measurements, and on 3 occasions during a 6-month trial, and patients didn't necessarily sign up for this study. So we went to a little bit of easier approach, but also to my colleagues in India. And this was done with Dr. Rajiv Mehta, who's in Surat. And we have a publication just came out in the World Journal of Gastroenterology that is a very pilot-like study, and it was open labeled. But all I wanted to show is that there is improvement in symptoms, analgesic use, and global health status in patients who take simvastatin. So what do these bars represent? So the first set are.
Excuse me, Stephen, for interrupting you. You were already 10 minutes over the time, so you can write it up. So make the conclusions.
So the conclusion are that we're entering a new stage of improved treatments for pancreatic diseases. And I was trying to show some examples of where we are in the process. Thank you.
Thank you, Steve. We will have room for one question. Dr. Saluja. Thank you. Excellent presentation.
Hello, Steve. I wish you were here, but good to see you online. Very nice presentation in different areas. Great. I just have a quick question about your work on somatostatin and acute pancreatitis. So maybe you showed it and it's late and I might be taking a nap, but did you see any changes in trypsin activity in your animal models with the somatostatin?
Actually, good question. It's simvastatin, but the second thing is yes, definitely. We saw a dramatic increase in trypsin activation, and it follows what your work several years ago showed that that there is less accumulation of those dysfunctional autophagic vacuoles, and that gives you better trypsin degradation.
And any effect on inflammation?
Yes, definitely.
Things which drives at least acute pancreatitis. So
yes, trypsin. So actually, Miklos and I talk about this all the time. Trypsin is the driver of inflammation, we think. And yes, inflammation was decreased.
Thank you.
So hi, Steve. Peter, thank you very much for your very nice presentations. We are really sorry that you and the others couldn't join. Currently, we must close the sessions.